target protein on the end is what you're using for affinity to bind other proteins. Okay? So any protein that's in the cell that interacts with your protein, and if it interacts strongly enough, it'll bind to that. It'll bind to your protein. So then you bind to the first protein, which has IgG beads, which bind to the protein A, and then you elute off that column with a protease. So that protease cleaves a very specific place, but it will leave your enzyme alone, or protein alone, leaving the other proteins which are bound to your proteins alone. So that complex stays. Okay? So to purify that complex further, you have these calmodulin beads, which then binds that calmodulin binding peptide. So that when you end up here with this complex that has your protein and has the proteins here, which are shown there with these squares or sort of in plaid. Those are the proteins which bind to your protein normally. Okay? So this is the protein you're interested in. These are the proteins that bind to that protein normally in the cell. Okay? So you purified these proteins by using your protein as an affinity tag. Right? So what kind of information do you get out of this? So here we show that there's a tag and your bait. So the bait protein is, say, your protein that you're interested in, and then the bait also interacts with numerous proteins. In this case, this showed you're interacting with 5 other proteins in the cell. Okay? So after you purify those proteins which interact with the protein you're interested in, you can run those out in an STS page show that we talked about last class, and that STS page will separate those out by size. Okay? So here you have the bait, and the bait will have certain proteins bound to it. You take that complex once it's been purified, you run that out on an STS page gel. After you run that out on an STS page gel, you'll get bands of certain proteins, but you don't know what the identity of those proteins are. Okay? But then you can excise those bands. You can take those bands and cut them out of the gel, and then you can take those proteins and run them on mass spec, like Molly taught. And then through mass spec, the mass spec will give you a fingerprint of what that protein is, and you can identify the identity of those proteins. So you have a protein that you know, and you use this tandem affinity purification to purify proteins you don't know. Okay? And once you purify those proteins you don't know, you can send them through something like mass spec, and mass spec will tell you the identity of those proteins. Yes?
Does trypsin just like get rid of the polypropylene?
No, trypsin. So it's similar to say amino acid sequencing. Trypsin is just going to cut up in pieces, so it's easier to send through the mass spec.
So then, how can you send it through mass spec if it's in all this gel?
You have to purify it out of the gel. So you cut the band out of the gel, and then there's certain solutions that'll extract the protein from the gel. And then once you extract it from the gel, you add trypsin, and you take that and send it through the mass spec. Any other questions? All right, so another technique used to look at protein-protein interactions is something called the yeast 2 hybrid assay. Okay? The yeast 2 hybrid assay takes advantages of 2 things. One is transcription of reporter genes, and the transcription of reporter genes by a transcription factor. So if you have a gene, there's an upstream region, activation region that binds transcription factors. Right? You can take advantage of that transcription factor binding to transcribe a reporter gene to understand protein-protein interaction. And what happens is you have something that looks like this, where you have a reporter gene and you have a transcription factor. Down here, it's the transcription factor is called GAL4. And GAL4 has 2 regions. One is an activation region, up there called AD, and one's a binding region, binding domain, BD. BD binds to the DNA, AD recruits the RNA polymerase so that you can transcribe this gene. Those two domains do not have to be right next to each other. Okay? If you were to just add AD and BD by themselves, they would associate a good drive transcription. So what you do is you can, well, I first should say what a reporter gene is, right? So we've gone over what transcription is. Transcription is synthesis of messenger RNA. That messenger RNA eventually gets turned into protein, and that protein would then have some property within the cell or job to do. In this case, the reporter gene is, two most common reporter genes are LAC-Z, which is beta-galactosidase, which you can use to cleave specifically a chemical that makes a blue color. So anytime you have the presence of beta-galactosidase and you have this compound that produces blue color. So you know if the colonies or the cells that you're looking at are blue, you know that that gene must have been turned on. And then there's a gene called His3, which catalyzes the last step of histidine synthesis. So if this His3 is not working, your cells will only grow in the presence of histidine. So if you put cells that only grow in the presence of histidine, and you put them on media that does not have histidine, they'll only live if that gene gets turned on. Right? And that gene can only get turned on if proteins are interacting with each other. Okay? So in this picture here, this is the GAL4 binding domain, this is the GAL4 activation domain, and then through molecular biology techniques, you fused two proteins of interest, right? You put one protein on the binding domain, one protein on the GAL4 activation domain, and if those two proteins interact, they'll bring those two domains closer.